There's a seed that costs only $2 a bag, and according to its advocates, it does far more than simply manage fatty liver disease. The claim is not that it reduces symptoms, slows progression, or controls inflammation through lifelong medication. The claim is that it reverses the condition by triggering the liver's own repair systems at the molecular level. Supporters point to more than 1,000 peer-reviewed studies and describe it as the most powerful liver protective substance ever identified, whether natural or pharmaceutical. Physicians in the Mediterranean region reportedly used this seed for liver disorders nearly 2,000 years ago. Modern randomized controlled trials published in hepatology journals have documented reductions in liver enzyme measurements, decreases in hepatic fat accumulation, and stimulation of damaged liver cell regeneration. Millions of people receive a fatty liver diagnosis each year. At the same time, a vast industry has developed around managing the condition. The worldwide liver disease treatment market generates more than $25 billion annually. Prescription drugs, specialist appointments, liver monitoring programs, and referrals for bariatric surgery all focus on the consequences of liver disease. According to proponents of milk thistle, the central issue remains oxidative stress and the inflammatory processes that continue damaging liver tissue. They argue that as long as those mechanisms persist, the disease advances and treatment continues indefinitely. The seed in question comes from milk thistle. Advocates maintain that it directly addresses the cellular injury associated with fatty liver disease. They further argue that because the remedy is inexpensive and cannot be patented, it has never become a major commercial focus, despite its long history. Milk thistle, known scientifically as Silybum marianum, grows naturally throughout the Mediterranean region and is also found across Europe, parts of Asia, and North America. The plant is recognized by its purple blossoms and white-veined leaves. Historical records suggest that its medicinal significance has been acknowledged since antiquity. One of the earliest known references appears in the work of Dioscorides, the Greek military physician whose text, De Materia Medica, served as a leading pharmacological reference for roughly 15 centuries. Writing during the 1st century AD, he described the use of milk thistle for disorders involving the liver and bile system. He reported improvements in symptoms associated with liver illness, including yellowing of the skin, abdominal swelling, fatigue, and weakness. Pliny the Elder also mentioned milk thistle in natural history, describing its use for conditions Roman physicians associated with bile and liver dysfunction. In a society where liver disorders were common because of wine consumption, preserved foods, and exposure to mycotoxins, Roman practitioners accumulated generations of observations suggesting therapeutic value. Knowledge of the plant continued through the medieval era. Herbal practitioners from the 13th through the 17th centuries repeatedly referenced milk thistle for liver-related complaints. Hildegard of Bingen, the 12th century German abbess and polymath whose medical writings influenced Europe for centuries, recorded preparations made from the plant for liver ailments. Over nearly 2,000 years of European medical tradition, milk thistle became so strongly linked with liver health that the name Silybum itself became closely associated with concepts of hepatic protection within herbal pharmacology. A major development occurred in 1968 when researchers at the University of Munich identified and characterized the plant's principal active compounds. Professor Wagner and his team isolated what became known as silymarin. Silymarin is not a single chemical substance. Rather, it consists of a flavonolignan complex that includes silibinin, silidionin, and silicristin. These compounds are concentrated primarily within the seeds and fruit of the plant. Wagner's group published findings describing remarkable hepatoprotective effects. The results immediately attracted attention from pharmaceutical companies. Efforts began to create synthetic versions of silymarin that could be patented and commercialized. Those efforts produced limited success. Researchers found that the biological activity of silymarin depended on interactions among multiple compounds existing in specific natural proportions. Artificial alternatives were unable to duplicate the complete range of observed effects. Meanwhile, naturally derived silymarin extracted from inexpensive milk, thistle seeds, could not be patented. As a result, pharmaceutical development shifted toward other strategies. Milk thistle remained largely within the herbal supplement market. While scientific literature continued to expand around its potential applications, advocates argue that understanding milk thistle requires understanding fatty liver disease itself and the reasons treatment has proven challenging. Fatty liver disease, medically termed hepatic steatosis, develops when more than 5% of liver cells contain excessive fat deposits. During the early phase, known as non-alcoholic fatty liver disease, or NAFLD, this accumulation can be reversed. A more advanced stage, non-alcoholic steatohepatitis, or NASH, occurs when fat buildup initiates persistent inflammation that damages liver cells. Over time, NASH can progress to fibrosis, a process in which scar tissue replaces functioning liver tissue. Fibrosis may eventually advance to cirrhosis. Cirrhosis is considered irreversible. The mechanism driving this progression centers on oxidative stress. When liver cells become overloaded with fat, they generate excessive amounts of reactive oxygen species. These unstable molecules damage cellular membranes, mitochondria, DNA, and protein structures. As injury accumulates, the immune system responds. Immune cells enter liver tissue, chronic inflammation develops, and inflammatory cytokines activate stellate cells responsible for scar tissue production. Fibrosis then begins to form. Pharmaceutical research has attempted to interrupt specific stages of this process. Anti-inflammatory drugs have been developed to suppress immune activity. Insulin-sensitizing medications have been designed to reduce the triggers associated with fat accumulation. Antifibrotic compounds have been investigated in hopes of slowing scar formation. Yet, after decades of clinical trials, not a single pharmaceutical medication had received approval for NASH treatment in the United States, according to the source material. Trial after trial failed to demonstrate the level of effectiveness and safety required for regulatory approval. While tens of millions of individuals continue living with the progression of fatty liver disease, a pharmaceutical answer remains absent. Meanwhile, milk thistle, supported by more than 1,000 studies and investigated for 50 years, remains available as a supplement. According to the source material, silymarin works through 5 concurrent biological actions that target the entire sequence involved in fatty liver disease progression. The first mechanism involves antioxidant activity. Silymarin is described as one of the strongest naturally occurring antioxidants identified for liver tissue. It neutralizes reactive oxygen species responsible for initiating cellular injury in fat-laden liver cells. Its role extends beyond removing existing reactive oxygen species. It also increases the activity of the liver's own antioxidant defenses. These include superoxide dismutase, catalase, and glutathione peroxidase. Those enzymes serve as part of the liver's protective system against oxidative injury. By enhancing their activity, silymarin reduces oxidative stress at the source of the disease process. The second mechanism involves protection of liver cell membranes. Silymarin becomes incorporated into the phospholipid bilayer of hepatic cell membranes, strengthening their structure. With stronger membranes, reactive oxygen species encounter greater resistance when attempting to cause damage. Toxic substances entering the liver also face increased difficulty penetrating liver cells. The source material states that this property explains why silymarin has been used in European emergency medicine as a treatment for poisoning caused by amanita mushrooms, among the fastest-acting lethal liver toxins known. German hospitals administer intravenous silymarin to patients suffering from mushroom poisoning. According to the source, this intervention has saved lives. The third mechanism centers on reducing hepatic inflammation. Silymarin suppresses the NF-κB inflammatory pathway. NF-κB functions as a primary regulator of inflammatory gene expression within liver cells. Once activated, it initiates the expression of hundreds of genes responsible for producing inflammatory substances. Silymarin interferes with this activation process. As a result, the inflammatory sequence associated with liver cell destruction is interrupted at what the source describes as its genetic control center. The fourth mechanism concerns stellate cells. These liver cells generate fibrotic scar tissue when inflammation is present. By lowering inflammation and directly suppressing stellate cell activation, silymarin slows the development of fibrosis. According to the source, during early-stage disease, it may also reverse this process. The fifth mechanism involves regeneration. The liver possesses an unusual ability to restore lost tissue and can regrow from as little as 25% of its original mass. Silymarin is said to enhance that regenerative capability by stimulating ribosomal RNA synthesis in surviving liver cells. Cells that would otherwise remain inactive begin dividing and replacing damaged tissue. A meta-analysis published in the United European Gastroenterology Journal during 2017 reviewed all available randomized controlled trials involving silymarin treatment for NAFLD and NASH. The analysis reported significant decreases in liver enzyme levels, hepatic fat accumulation, and inflammatory markers among patients receiving silymarin compared with those receiving placebo. According to the source material, no pharmaceutical treatment for NASH had achieved comparable outcomes in a completed randomized controlled trial. Milk thistle, derived from a $2 bag of seeds, repeatedly demonstrated these findings. The source then shifts to the economic and regulatory environment surrounding treatment development. More than 1 billion people worldwide are estimated to have some form of fatty liver disease. It is described as the most common liver disorder throughout the developed world. The global market for liver disease treatment generates more than $25 billion each year. Research aimed at developing NASH therapies has ranked among the most heavily financed sectors in pharmaceutical development for the past 20 years. Dozens of drug candidates have advanced into clinical trials targeting NASH. According to the source, none achieved regulatory approval. The source argues that the reason is not that the disease lacks a treatment, but that silymarin itself cannot be patented. The economic reasoning presented is straightforward. A patented pharmaceutical product for NASH could potentially be sold for hundreds of dollars each month. With 1 billion affected individuals requiring ongoing treatment, the resulting revenue opportunity could reach tens of billions of dollars annually. A $2 bag of milk thistle seeds, ground and consumed daily by patients, represents a different economic model. The source characterizes it as a challenge to the financial assumptions underlying NASH drug development. According to this argument, investment continued flowing toward unsuccessful pharmaceutical trials rather than toward a compound that had existed in Mediterranean fields for 2,000 years. Natural silymarin cannot receive patent protection. The milk thistle plant has grown throughout Europe and the Mediterranean region for millions of years. Because no organization can obtain exclusive ownership rights over Silybum marianum, there's little financial motivation to fund the large Phase 3 clinical studies necessary for regulatory approval of milk thistle as a NASH therapy. Without that approval, milk thistle products cannot advertise therapeutic claims. They are marketed as dietary supplements and limited to general statements regarding liver support. According to the source, manufacturers cannot state that more than 1,000 peer-reviewed studies support reversal of hepatic steatosis. They cannot claim inhibition of NFKB activity. They cannot claim reduction of hepatic fibrosis. They also cannot describe silymarin as the most extensively studied hepatoprotective compound in medical history without the regulatory approval process that requires clinical trials costing hundreds of millions of dollars. The source notes that companies selling a $2 product cannot finance clinical programs costing hundreds of millions. At the same time, pharmaceutical corporations capable of funding such trials have little incentive to prove that an inexpensive supplement addresses a condition they hope to treat with a drug costing $200 per month. The source characterizes this situation not as a conspiracy, but as the structure of a regulatory framework that leaves natural remedies largely out of view. Even so, the knowledge persisted. It remained present within European herbal medicine, integrative hepatology, and an expanding body of peer-reviewed research that has made silymarin one of the most extensively investigated natural substances in medical literature. In Germany, milk thistle extract standardized for silymarin content is approved as a pharmaceutical medication for liver disorders rather than being classified solely as a dietary supplement. German physicians prescribe it for toxic liver injury, chronic hepatitis, and cirrhotic liver disease. It is reimbursed through German health insurance. The source emphasizes that a substance unable to make therapeutic claims in the United States is prescribed as a pharmaceutical product within one of the world's most scientifically rigorous healthcare systems. A randomized controlled trial published in the World Journal of Gastroenterology during 2015 enrolled patients diagnosed with NAFLD. Participants were randomly assigned to receive either silymarin supplementation or placebo for 8 weeks. At the conclusion of the study, the silymarin group demonstrated significant reductions in ALT and AST, the primary liver enzyme indicators associated with hepatic cellular injury. Ultrasound evaluations of hepatic fat content also showed significant improvement among individuals receiving silymarin. The placebo group showed no significant changes. A meta-analysis published in the European Journal of Gastroenterology and Hepatology during 2020 reviewed 12 randomized controlled trials examining silymarin in NAFLD. Across all 12 studies, silymarin was associated with statistically significant reductions in liver enzyme measurements, hepatic fat accumulation, and inflammatory markers. The authors reported that the body of evidence supporting silymarin for NAFLD exceeded the evidence available for any pharmaceutical compound evaluated for the same purpose. An integrative hepatologist practicing in Switzerland has included milk thistle-based protocols in a fatty liver program since 2009. According to the source, patients diagnosed with NAFLD who consistently followed the protocol for 6 months demonstrated measurable improvements in liver enzyme values during follow-up laboratory testing. Ultrasound examinations documented reductions in hepatic fat content among most individuals who adhered to the protocol. The reported outcomes aligned with findings described in published research. The source argues that the underlying chemistry remains unchanged regardless of whether the FDA has authorized therapeutic claims. The protocol described by the source begins with obtaining milk thistle extract standardized to 80% silymarin. Standardization is presented as important because raw milk thistle seeds and non-standardized preparations may contain highly variable concentrations. Most clinical studies utilized extracts with documented silymarin levels. The recommended target is 80% silymarin, the concentration reportedly used throughout the majority of research. A bottle containing 60 capsules of standardized milk thistle extract at 80% silymarin is described as costing between $8 and $18. At a dosage of 2 capsules each day, the supply lasts roughly 1 month. The estimated daily expense ranges from approximately 25 cents to 60 cents. The second step involves consuming 300 to 600 milligrams of silymarin per day, divided into morning and evening doses. According to the source, studies reporting significant improvements in liver enzymes and hepatic fat content generally used doses within this range. The recommendation is to begin with 300 milligrams and evaluate tolerance over 2 weeks before increasing the amount if needed. Most users reportedly tolerate milk thistle well. Documented adverse effects are described as uncommon and generally mild, with occasional loose stools reported in some people taking higher doses. The third step is taking the supplement with food. Silymarin absorption is said to improve in the presence of dietary fat. Consuming the extract during a meal containing some fat may increase bioavailability. Examples mentioned include breakfast or dinner accompanied by olive oil, nuts, or avocado. The fourth step addresses dietary contributors associated with fat accumulation in the liver. According to the source, milk thistle helps reverse existing damage and protect against additional injury. However, continued intake of the dietary factors linked to fatty liver, especially excess fructose from added sugars and refined carbohydrates, may work against the supplement's effects. The recommendation does not call for a strict eating plan. Instead, reducing added sugar intake and increasing vegetable consumption are described as measures that decrease hepatic fat accumulation. The source states that combining milk thistle supplementation with lower sugar intake produces faster results than either approach alone. The fifth step focuses on consistency. The source recommends maintaining the protocol for at least 3 months before evaluating progress through blood testing. Normalization of liver enzymes and reduction of hepatic fat are described as cumulative processes. Clinical trials demonstrating meaningful improvements generally lasted from 8 to 24 weeks. Patients are encouraged to ask their physician to include ALT, AST, and GGT in their next blood panel. These markers are commonly elevated in fatty liver disease and may return toward normal levels as the condition improves. Recording these values at baseline, then again after 3 months and 6 months, is presented as a way to objectively track changes over time. The timeline described by the source begins during weeks 1 through 4. During this period, silymarin accumulates within liver tissue. Antioxidant enzyme systems become increasingly active and oxidative stress begins to decline. No visible or symptomatic changes are expected at this stage. Between weeks 4 and 8, liver enzyme values may begin decreasing on blood tests. Hepatic inflammation is described as declining. Some users reportedly notice improved energy levels and less discomfort in the right upper quadrant. By weeks 8 through 12, statistically significant improvements in liver enzyme measurements are described as typical. Ultrasound examinations may also reveal measurable reductions in hepatic fat content. Months 3 through 6 represent the endpoint used in much of the published research. At this stage, hepatic fat levels are reported as significantly reduced. Liver enzyme values approach the normal range. In early stage NAFLD without substantial fibrosis, multiple clinical trials have documented disease reversal during this time frame according to the source material. The estimated total cost of a 6-month protocol is approximately $50 to $100 for 6 months of standardized milk thistle extract. The source compares that amount with 6 months of hepatology consultations costing between $200 and $400 per visit. Those appointments may include monitoring disease progression and recommending lifestyle modifications, while no approved pharmaceutical treatment exists for the condition according to the source. The argument presented is that a supplement costing $50 to $100 possesses more clinical evidence than any pharmaceutical product tested for the same indication. The source concludes by revisiting its historical timeline. Dioscorides described the remedy during the 1st century. Medieval herbal practitioners continued using it across 10 centuries of practice. German pharmaceutical authorities later approved it as a prescription medication. More than 1,000 peer-reviewed studies have examined it. Yet in the United States, milk thistle remains available on supplement shelves without therapeutic claims. The source notes that the silymarin contained within milk thistle seeds carries no patent. The inhibition of NF-κB attributed to the compound requires no prescription. The liver cell regeneration associated with it creates no billing code. According to the source, fatty liver disease has been reversible for 2,000 years through a seed costing $2. The claim is that the underlying biochemistry has not changed. Instead, the source argues that regulatory and economic structures have developed around the assumption that a patentable pharmaceutical answer is necessary, despite such a treatment not yet existing. Today, approximately 1 billion people worldwide are estimated to have fatty liver disease. The pharmaceutical industry has devoted 20 years and hundreds of billions of dollars toward developing a drug for the condition. According to the source, the proposed solution has existed in Mediterranean fields since before the Roman Empire. The source concludes that this situation is not evidence of conspiracy, but rather the result of economic incentives within an industry that cannot generate substantial profit from a $2 seed. If this information gave you a new perspective on fatty liver disease and the long history of milk thistle, share this discussion with others and continue exploring the research for yourself. Staying informed remains one of the most valuable tools in understanding any health topic. Please leave your thoughts about this content in the comments below. Thanks for watching. See you in the next one.